Breakthrough in neuroscience. Can our brains really create new neurons in adulthood? Researchers have made a groundbreaking discovery that could definitively answer one of the most debated questions in neuroscience: whether the adult human brain can continue to form new neurons. This discovery puts to rest years of debate and confusion, potentially changing how we view brain function and regeneration. The phenomenon known as neurogenesis occurs in the hippocampus, an essential brain area that plays a pivotal role in learning, memory, and emotional regulation. Put simply, our findings settled the long-standing debate on whether adult brains can generate new neurons, explained Marta Patalini, co-lead author of the study and researcher at the Karolinska Institute in Stockholm. We've gathered compelling evidence that supports the theory of neurogenesis in adults. Experts across the field are in agreement, highlighting that this research provides substantial support for the existence and proliferation of stem cells and neuron precursors throughout adult life. Though a single study alone isn't conclusive proof, this represents a strong case validating the idea that our brains contain active cells capable of generating new neurons. But here's where it gets controversial. The identification of new brain cells hinges on the quality of brain samples, something that has historically been very challenging with adult human tissue. The handling and preparation of human brain samples can sometimes obscure or hide new neurons, Patalini noted. Thanks to innovative technologies and methods, the team managed to navigate these hurdles. The researchers analysed over 400,000 individual nuclei of hippocampus cells from a diverse age range, including both children and adults, combining advanced imaging and machine learning techniques to pinpoint where new cells were located within the tissue. Contrary to what some might expect, new dividing precursor cells weren't limited to infants. They were also detected in teenagers and adults. What really stands out here is that new cells were found not only in children's brains, but also in adults. These precursor cells have the ability to self-renew and generate other brain cells, Patalini elaborated. And this is the part most people miss. With the use of fluorescent tags and sophisticated algorithms, the team could identify which cells were likely to become neurogenic stem cells, a tactic that dramatically improved the accuracy of their findings. Though children's brains naturally produce a higher rate of new neurons, about 9 out of 14 adult brains reveal signs of neurogenesis using one method, whereas every adult brain analysed with another method showed evidence of newly formed cells. Still, the variance in some adult brains without new cells remains a mystery that requires more exploration. In the next phase, researchers might investigate whether the generation of new brain cells in adults is a response to neurological conditions like Alzheimer's, or if it represents a marker of robust brain health. Catching these needles in a haystack opens up opportunities for understanding how these precursor cells might relate to disease and longevity, said Dr. W. Taylor Kimberly, a neurocritical care expert at Massachusetts General Brigham who wasn't involved in the study. Controversial thought. Could the presence of new neurons actually be connected to enhanced cognitive resilience in aging adults? Does this discovery pave the way for future therapies that could harness these natural brain processes? Share your thoughts. Do you believe this connection could lead to breakthroughs in treating brain-related illnesses, and could it redefine our approach to brain health? Leave your comments below.